We're here with Dr. Luwy Musey, who is Executive Director in Biologics for Vaccine Clinical Research at Merck Research Laboratories. We also have Dr. Stephen Pelton, who is an senior attending physician of pediatric infectious diseases. Dr. Pelton, could you give me just a brief background on yourself, and then we'll move on to Dr. Musey?
Sure. I've been at Boston Medical Center, which was Boston City Hospital before that, I actually started my 49th year this week. I have been acting chairman. I've been the chief of pediatric infectious diseases. And my main focus and interest has been in vaccine preventable diseases for the last 25 years. And throughout my career, it's been clear how important pneumococci are and the burden of disease in children and in adults caused by pneumococci. When we look at, say, the turn of the century in 2000, strep pneumo was the number one cause of bacterial meningitis in children. It was an important cause of sepsis and pneumonia, and it's a global problem. We have more than 500,000 deaths a year on a global basis in children under the age of 5 due to strep pneumo. And it's amazing how much progress we've made. I would say in my career, there have been 3 events that have been sentinel in the progress we've made in preventing serious disease in children. 1, the prevention of HIV transmission from mother to child, and 2, the treatment and the conversion of HIV from a fatal disease to a chronic disease. 2, the introduction of Hib vaccine, Haemophilus influenza type B, which I used to see a case a week. Now I haven't seen a case of Haemophilus type B disease in at least 5 years. And then strep pneumo, the pneumococcal conjugate vaccines, which took and made a severe disease, a life-threatening disease, sepsis, bacterial meningitis, into a relatively rare condition now in the United States.
Thank you for that introduction, both to yourself and the topic of conversation today. Dr. Musey, would you like to do the same?
Yeah, so I'm Luwy Musey. I'm a physician as well. I've been at Merck for 19 years now. I work in vaccines all the time. And I've been also impacted by disease like Dr. Pelton. My youngest sister had measles in front of me when she was less than 1 year of age. And so developing vaccine to me has always been something that is very dear to me. Since I've been at Merck, I was involved in many different vaccines like measles, mumps, and rubella. And I had the wonderful privilege to meet with Dr. Maurice Hilleman as I joined Merck. And so for me, it's really continuing that heritage started by Dr. Hilleman and continuing today. And developing the pneumococcal vaccine, I've been on the program for 11 years. And this has been really the most important work that I've been doing so that we can provide a best vaccine possible that can really prevent pneumococcal disease and save lives.
So on that subject of making that new development in the vaccinology here, Dr. Pelton, could you introduce a bit of where obviously we've made a lot of progress with the current set of PCV we have, but where are some of the pitfalls or what do we need to improve vaccinology and the strategy there in terms of serotypes, what's circulating?
So I think there are 2 main challenges for moving forward. One is within the current vaccines that we have, PCV10, which is not a U.S. product, and PCV13, which is widely used in the U.S., and about 90% of children have received that. We have 1 or 2 serotypes where our protection is not as good as we would like it to be, and that's especially true for serotype 3, which is an important cause of severe pneumonia in children and adults. And then what we've also seen is that over time, there's been a shift in the serotypes that cause disease, especially in the adult population, but also in children, but especially in people who have comorbid conditions, those that have asthma, diabetes, lung disease, heart disease, or even more severe conditions like HIV or immune deficiency or cancer. And these serotypes probably are a little bit less virulent than the serotypes that were in the 13-valent vaccine, but they are certainly able in the right host to produce severe disease. And the right host appears to be either children whose immune system are not fully developed, or children who have comorbid conditions, which means that their ability to fight infection is somehow hampered, or the adult population, especially over 65, where there's aging of the immune system and weakening of the immune system, and therefore susceptible to these maybe less virulent vaccine, what we call now non-vaccine serotypes.
Dr. Musey, can you speak to anything with Merck's pipeline here in terms of the three that we're sitting down and talking about today in terms of addressing those needs?
Yes. So before I go there, I just want to talk about something that is very important, is that Merck has been involved in the development of pneumococcal vaccines since the 70s. And so, there's been study with the 6-valent vaccine and 14-valent vaccine, and now the 23-valent vaccine, polysaccharide vaccine, that really have shown that the vaccine is really cost-effective. And so there are data that shows that Pneumovax 23 is really the vaccine that is really good for the adult and can really prevent cause of pneumococcal disease. But we also understand that Pneumovax 23 has limitation when it comes to disease in children below the age of 2. That is why Merck decided in the early 2000s to start developing another vaccine. And when we did that, we did exactly what Dr. Pelton said. Try to look at what exactly are the issues. The two issues are, number one, when you develop a vaccine, you need to make sure that the new vaccine does provide the same level of protection as the other vaccine that was previously licensed. So when we were thinking about our vaccine, knowing that we'll be coming after Prevnar 13, we wanted to make sure that we are providing the same quality of immune response, if not better, than Prevnar 13. And so that's what you see in our studies. And because of that, we spent 4 additional years to really delay our program to make sure that we are developing the best vaccine possible. The second issue beyond the maintenance or the suppression of disease is trying to really look at the science, the epidemiology, that is going to teach you, like Dr. Pelton said, what are the serotypes that are emerging and that are causing disease that are not in the vaccine. And when we looked at the serotype that we have selected, among the non-vaccine serotype, 22F and 33F are among the leading cause of pneumococcal disease in both adults and children. So that's number 2. Number 3, and when you look into that, you learn about the science about how do you make a vaccine. And we've learned a lot making VIR-4. That really now is enabling us to say, what is the next step beyond VIR-4? And so we have a very broad pipeline with looking at the disease burden that is very specific to the adult. The residual disease is much important, that caused by the non-vaccine type in adult and in the children. So we are using a different approach that is tailored specific to the adult and to the children. So overall, our portfolio of pneumococcal vaccine is really looking at what's happening today with VIR-4 and also what's going to happen in the near future. So that is the approach that Merck is taking to make sure that we are really addressing what we call the unmet medical need that is tailored to each population. In addition to that, give me 1 second, for the VIR-4, we also made sure that our clinical development program is not just about getting licensed. It's about studying the population that needs the vaccine the most. What that mean? That means that you study people who are at increased risk for pneumococcal disease, either due to age, the very young and the very old, but also the population Dr. Pelton mentioned, those who are at increased risk, and those are at high risk. So we have studies in HIV, in stem cell transplant. We also have studies that involve people who have chronic heart, chronic lung, chronic liver, diabetes, asthma, and so forth. We also include in that study Native Americans in the study. Why? Because there's a population that's really also had increased pneumococcal disease. So our vaccine development really is tailored to really the population that need the vaccine the most.
Excellent answer. And to just go back to what you were saying initially with your first point, has there been, I know there's phase 3 initial data, has there been things pointing towards the non-inferiority you were describing?
Yeah, so at the moment, the protocol 18 that we show about HR in the HIV population and the protocol, that was really the protocol 18 was really about HIV population was not tailored really to show, there was not an hypothesis to show non-inferiority to Prevnar 13. And protocol 21, the flu study, was not a study that had a Prevnar 13 as a comparator. However, we have data from our phase 2 study, protocol 6. In that study, we compare side by side V14 and PCV13 in adults 50 years of age and older who are pneumococcal vaccine naive. And if you look at our data, we've accomplished exactly what we said, which is, number one, making sure that we maintain the same level of response that we have compared to Prevnar 13 for the 13 serotype. And if you look at the GMT ratio, the OPA-GMT ratio, because we measure by OPA, which is an antibody that allows for the killing of the bacteria, the GMT ratio across all 13 serotypes is 1.19. So that means that we are doing as well, if not better than Prevnar 13 for that. In addition, like Dr. Pelton said, we have greater immune response to serotype 3, which we did not formally test as an hypothesis, but that has been consistent across all our studies. So we're addressing the issue with serotype 3. Number 3, as we have contained or achieved our goal of maintaining the same level of response, even better for serotype 3, now we added 2 serotypes, 22F and 33F, for which we show we have good response. We are convinced that our data in our pivotal study, protocol 19, which will be coming pretty shortly, will really confirm those data from protocol 6, which is really to show that we are making sure that immune response, the quality of immune response is very maintained before thinking about adding new serotypes. Because the 13 serotypes that in Prevnar 13 were among the serotypes that were causing a great deal of disease. There was a reason why they would pick among the 13 first. So trying to lower the guard on those 13 serotypes could be very damaging because those serotypes were very invasive and they were also associated with antibiotic resistance.
Absolutely. And just to kick things back to Dr. Pelton for a moment, I wanted to ask what the global burden looks like. I mean, I know a bit of it myself, but if you could give us your kind of expertise on the global burden, and then we can hear a little bit about what this might do to impact as it continues to show these promising results.
So the global burden in the world is mostly targeted at the developing world, where disease is much more common in the African continent, in India, in parts of Indonesia. And the biggest illness, or the major form of illness there, is pneumonia. And pneumonia is a regular killer of children. And unfortunately, we have made a lot of progress with the vaccines we have, but the serotype distributions in different parts of the world are slightly different. They're not all, the original vaccine, PCV7, was designed based on what's common in North American children, US, Canada, and Mexico primarily, and did not cover all the serotypes. And then when we went to a 13-valent, it was a more global product that had serotypes that were uncommon in the US, but common across the globe. It also had important serotypes for the US because 19A had become antibiotic resistant and had emerged as a major cause of disease in the US. So it addressed both. And the new vaccines will address, again, important serotypes. If we look at 22F and 33F, certainly in Europe and the United States, they are two of the most important non-vaccine serotypes. In other parts of the world, they're in the top 10 maybe, but maybe not the most important or the most 2 or 3 most important. And I think that, again, this is why this is an evolution. It's not a complete answer. We will need vaccines that address larger numbers of serotypes in the future, or we need what is referred to in the pneumococcal world as the Holy Grail, which is a vaccine that is protein-based, that addresses all pneumococci, but like the Holy Grail, we've yet to find it yet. And there's not a likelihood in my mind that we will find it, but it's out there as a hope.
Right. And I mean, too, when you think about something like HIV cure, the definition has changed over time. And so, we may wake up one day and find ourselves with a very surprising pneumococcal solution. But in the meantime, right, we've got to work with what we have and these evolutions over time. And so I guess my only question with that is, what exactly is the situation with, does it lessen the impact or lessen the immune response to pile on more serotypes, for example?
It's a terrific question because you might say, well, more is better, so just keep adding more. However, what we've learned as we've gone from 7-valent to 10-valent to 13-valent to the future, 15 or 20, is that as we expand the number of serotypes, if we're not cautious, we reduce the immunogenicity of each serotype. And of course, the concern is, as Luwy nicely talked about, is that if we impact the immunogenicity too much, some of the old serotypes may reemerge as problems. So one, we have to maintain the immunogenicity against what we already have. And two, the new serotypes, we don't know exactly how much antibody is necessary to prevent disease. We have a correlative protection, which is based on an average. It's called 0.35 micrograms per mil. But for any specific serotype, we don't know if 0.35 is enough. And we already have learned mostly, like for serotype 3, 0.35 is not enough. So we need not only to develop the studies to look at the immunogenicity, but then once the vaccine is introduced, we need to look and see, does it do what we expect it to do, prevent disease to the new serotypes that are in the vaccine, and again, keep enough immunogenicity so that we don't see reemergence of any of the old serotypes.
And I think that's actually a great springboard to kick things back to Dr. Musey. How, given you addressed some of that concern in the beginning, really, how does this, or these, I should say, vaccines, how do they come together to form that strategy that hits that right balance, that kind of Goldilocks zone of not damaging the immunogenicity?
Yeah. So, and as I said earlier, it's really learning from what exactly the science is telling you. It's learning about the science when it comes to what do we know about the sugars? How do we know about to put together the protein and the sugar? How do you know about the body, the immune system? How the immune system respond to that? And things have evolved. I mean, this is something that we have to keep up with what's happening in out there. And one lesson that we have learned very, very vividly for me is that we were doing the Viron-4 development. And then at some point, we were focusing mostly on the pediatric. But as we were listening to others, scientists, we learned that some scientists do favor, like the conjugate vaccine. So we said, let's offer the conjugate vaccine with Viron-4 for adults as well. And we generate first, because the adults only need one shot. So you get the data with one shot. And then the pediatric, you give multiple doses. But the key point is really the first 3 doses, when the kids get it at 2, 4, and 6 months of age, because the greatest burden disease for the children Dr. Pelton may not mention that, is really in children below the age of 1. It's before the child reached the age of 1. So it's very important to measure the immune response after the first primary series in infants. What we found was that the adult, they respond very beautifully to our vaccine. We were very pleased with that. But the children did not. Our vaccine did not meet what we saw in the adults. So 2 options: either start going back from scratch, fine-tune a little bit, or just continue with that particular first formulation. Merck made the decision not to continue with a vaccine that will not make a difference or will go against what we are talking about here, maintaining suppression of disease caused by the 13 serotype. So that's delayed our vaccine filing by almost 4 years, because we have to do phase 1, we have to do phase 2 in infants and adults, and now we're starting phase 3. And because of that, we looked at our data from the adults. We show you that protocol 6 for phase 2, and for the infant, protocol 8. You'll see that that particular goal has been achieved with V1-4. And we really are very determined to continue to keep the suppression as we think about future generation. And what we have learned in making V1-4 is helping us making the next generation. So when we talk about, Dr. Pelton talk about the adults, it's very specific. The size of the disease caused by the non-vaccine type is very different. Also, we know that in the next 20 years, the population above 6 years of age will double. And then more importantly, we know that pneumococcal pneumonia in adults costs billions of dollars in healthcare costs worldwide. So by looking at that, we felt that the adults themselves have a very unique unmet medical need. So rather than giving a vaccine in the future, they will cover both the adult and the peds at the same time, we decided to look at a vaccine that really would be tailored for the adults' needs. And we know for the pediatric, because of the indirect effect that the pediatric gives to the adults, the pediatric need also to be protected against emerging serotypes. So our V1-7 will have to continue with expanding so that we can get a better protections against disease caused by those serotypes, while still maintaining the suppression. And so we will not compromise on the science of maintaining the same level of response than Prevnar 7 and Prevnar 13, so that we can continue to maintain the suppression of the disease. So this is why we believe, and we also believe in some markets, our Pneumovax Trinity will still continue to be used because it has a great value, broad serotype composition, the cost is very low, and a lot of market will continue to use Pneumovax Trinity. So this is our strategy, and we are in V1-4 today, and we think this is a great value, especially, like Dr. Pelton said, if you really want to maintain the level of response, those subjects at increased risk, those persons at high risk, need to have a vaccine that will have this very strong immune response. And so we are studying those people in V1-4 so that in the future, when we think about different options, we are maintaining the level of response that we see in that population at increased risk and at high risk. So this is the value of V1-4, both age group, very young and very old, but also looking into those who are at increased risk, those who are at high risk, and maintaining the level of immune response that we have while looking at additional serotype. But you cannot compromise in saying, let me just add more serotype at the expense of 19A or serotype 3 or serotype 6B, for example, or even 19F, which is re-emerging now.